My name is Emmanuel Lozada Soto. I'm a PhD candidate at North Carolina State University. It is my pleasure to be here and share a little bit of my work throughout my PhD. And I will be talking about the actual status of the genetic diversity in 5 North American dairy cattle breeds. So, as it has been said previously, genomic selection has revolutionized livestock breeding. Martino Cassandro showed, in the Italian population, it has been extremely successful, and in the American population, in the USA, genomic selection has been incredibly successful at increasing the rate of gain for many dairy cattle traits, including those of low heritability, which is the great promise of genomic selection. And this has been done mainly by increasing the accuracy of prediction and decreasing these generation intervals. So, genomic selection has brought an indirect effect of faster accumulation of homozygosity. And this has been seen in Australian population. Beth Scott published a paper on that. It's been in Italian population, Michela Blondi, in Dutch populations, French populations. Overall, worldwide, the implementation of the implementation of genomic selection has brought on this detrimental accumulation of homozygosity. But in this talk, I will share the actual status in the US population, and more importantly, I'm going to talk to you about methods that can be utilized to monitor this accumulation of inbreeding. So what are the topics for my talk today are 3. First, I will show some results about the characterization of genome-wide and region-specific autozygosity. Secondly, I will show metrics of pedigree and genomic inbreeding and how these differ and how the trends differ between these in each breed. And lastly, this thing is a very important topic to hit on for today, is understanding these historical and recent changes in genetic diversity that have occurred in the US population of dairy cattle. So I would like to introduce you to the 5 genomic breeds of US dairy cattle, right? We have the Ayrshire, which originated in Scotland, the Brown Swiss originated in Switzerland, the Guernsey and the Jersey originated in the Channel Islands off the coast of Britain and France, and lastly the Holstein, which is a Dutch breed. All of these breeds have been around in one way or another since the late 1900s in the US soil. So they have some history in the US, and they have become the predominant dairy cattle breeds in the US. So my study, these are the animals that I have included. We were very fortunate to get access to over 4 million genotypes and pedigree data for all these 5 breeds, including 4 million Holstein, 500,000 Jerseys, 50,000 Brown Swiss, 10,000 Ayrshires, and around 5,000 Guernseys. Here, I present the number of genotyped animals by the year of birth for each breed. And you can see that with the implementation of genomic selection, the need for, and also the cost of genotyping has decreased substantially. We see more and more animals genotyped. So that is very good. So, to answer my first question of characterizing genome-wide and region-specific autozygosity, I first detected runs of homozygosity in these breeds. I use Plink, and I use the parameters shown here to detect the outer age. Here is the first results. These are some descriptive statistics for the runs of homozygosity that I detected. In the table at the top, I have the statistics for the number of ROH. We see some variation between the breeds, ranging from 37.24 runs in the average for the Ayrshires, all the way to 62.66 runs for the Jersey on average. On the bottom, we see the average ROH length, and we can, like Christian mentioned, this is related to the age of the inbreeding, and the average ROH length varied from 7.04 all the way from 6.80 from 6.74 all the way to 7.54 average RH length. But we can even break this down even more. In this plot, I present the descriptive statistics for the average number of RH, but I have broken down the runs of homozygosity into 4 categories based on the length. Those from 2 to 4 megabases, 4 to 8, 8 to 16, and 16 megabases or longer. We can see in this plot that the majority, for most breeds, the majority of the RH present in the genome are of the smaller length. So they presumably have been acquired in a more distant past, and through their combination, they have been broken down into the lengths that we see now. And we can see that these breeds, the Channel Island breeds, the Guernsey and the Jersey, they have a disproportionate amount of this short RH. So this is very indicative of ancient inbreeding in these 2 breeds. Conversely, in this plot, I show the average combined length of the genome in RH, and I also have the 5 breeds, and I have the 4 categories. And in this plot, you can see that the trend is towards average to long RH, being the combined length of moderate to large RH make up the majority of the genome. So you see that when you calculate this inbreeding coefficient based on the runs of homozygosity, you will have a proportion that is very that is large. That is this inbreeding due to pretty recent inbreeding. Also, with the runs of homozygosity, we have the opportunity to go even further into the genome and characterize region-specific autozygosity in opposition to these genome-wide measures. So to do this, I calculated a coefficient of autozygosity for every marker in the genome in all the 5 breeds, and it is the proportion of animals in which this marker was located inside a run of homozygosity. Then, we get these autozygosity measures. To detect, to classify SNPs as significant, I set the threshold at 99.5th percentile of all genome-wide markers for the autozygosity. Then I joined the significant markers that were adjacent into larger regions of homozygosity called runs of RH islands. And these are indicative of past selective history. So in these plots, I show for the 5 breeds Manhattan plots of the SNP-based autozygosity. We can see that there's many peaks around the genome for Ayrshire, Brown Swiss, and all the other breeds. But we were very interested into seeing, so these 5 breeds are independent, right? Do they have some shared history possibly? Can we find any regions that are shared between the breeds? And luckily we did. In the Ayrshire and in Brown Swiss, we found a 3.4 megabase region on chromosome 6 that contained these genes. These genes have are associated with these genes. These genes have been associated with components of milk and also health traits, such as clinical mastitis and ketosis. For the Holstein and Jersey, we will also find the overlapping region, this time on chromosome 20. This region was, this overlap was a little bit smaller at 2.6 megabases, and the genes that were located in the proximity inside this RH island, these overlapping RH islands, were the ISL1 and PILO gene, and these genes have been associated with reproductive performance, especially age at first calving, I think daughter pregnancy rate, and also maturity traits. So these are all in some ways related. I didn't show the results for the Guernsey because Guernsey, we didn't find any overlapping regions between the Guernsey and any other breeds. So that was an interesting result on itself. So to see if these regions of autozygosity are caused by selection, by selective pressure forced into these regions, we did an enrichment analysis where we annotated QTLs in these RH islands, and then we identified significantly enriched traits related to these QTLs that we annotated in these regions. These are the results for the Ayrshire and Brown Swiss. I remember I said that there was an overlapping region on chromosome 6, and we can see for the most part, these are the top 10 annotated traits based on the log 10, negative log 10 p-value. And we can see that many health traits and also production traits are significantly enriched in these breeds, right? Clinical mastitis being the top hit, and ketosis as well. Yeah. And also we see the milk production traits here and the milk composition. For the Holstein and the Jersey, we can see that there's also health traits such as dystocia, and there's also milk production traits, length of productive life, etc. So we see many dairy cattle traits being expressed in this analysis. And here are the results for the Guernsey. For the Guernsey, we had a very interesting result where many of the QTLs of the traits that we were significantly enriched were related to meat production, to meat and carcass. So that was a very interesting result in itself. We're not 100% sure why, but there is some bias in the QTL annotation in itself. So that might be an indicator of this, or some pleiotropic effect of the QTLs that are in charge of both dairy traits and meat and carcass. So to complete my second objective of this study, I calculated pedigree and genomic inbreeding coefficients. I calculated pedigree inbreeding using the PEDIC package and using up to 6 generations. And I also calculated genomic inbreeding using 2 very famous measures, using the genomic relationship matrix, using Van Raden's first method, and using 0.5 allele frequencies, which is the standard for calculating this inbreeding in this US dairy population. And we also calculated the proportion of the genome that contains RH, using where the RH identified were the ones I previously mentioned. So here are some results. In this first table, I have the within breed averages of the inbreeding coefficients for animals born between the year 2000 and the year 2020. We can see that for the genomic measure especially, there is a very big difference between the different breeds. But to summarize overall, we saw that the pedigree inbreeding ranged from 0.06 in Ayrshire and Brown Swiss to 0.08 in Holstein and Jersey. The inbreeding coefficient based on the genomic relationship matrix ranged from 0.22 in Holstein to all the way from 0.29 in Guernsey and Jersey. And lastly, the RH inbreeding coefficient ranged from 0.11 in Ayrshire all the way to 0.17 in Jersey. So we can see how Jersey is one of these breeds that has been accumulating homozygosity, and this is not a surprising result, and many different Jersey populations worldwide have seen similar trends. In the plot below, I show the same results, but by year of birth of the animal. So we see the trends in the average inbreeding by year of birth from animals born from the year 2020, all the way, 2000, all the way to the year 2020. First, I want you to notice how the different breeds, especially for these genomic measures, how the different breeds have different trends in inbreeding, and also how the accumulation of inbreeding has been disproportionate in the different breeds. For example, Holstein was one of the lowest, both in ROH and genomic relationship matrix in the year 2000, but the rate of accumulation of inbreeding has been staggering in both breeds. And now, for example, here you see Holstein overcoming the Ayrshire for the RH inbreeding coefficient. So it's very, very surprising. And also here in the Jersey, you see how Jersey, I showed in these results that have high inbreeding, but we see that this inbreeding probably was accumulated some time ago, and the Jersey breed has done a pretty good effort in maintaining this inbreeding, although we see some increases here during the early stages of the adoption of genomic selection. So we're not out of the woods yet. Here, I show the Pearson correlation coefficients, here heat map, and I show for the breed that had the lowest overall Pearson correlation between the different inbreeding coefficient measures. And the breed that had the highest. So Guernsey had the lowest and Holstein had the highest. You can see how pedigree inbreeding had moderate to high Pearson correlations with the genomic measures, and the two genomic measures had very high correlation between them, range from 0.95 all the way to 0.98. So we can see how even with pedigree inbreeding, we're capturing some of these genomic inbreeding that we're calculating with the genomic measures. So it's overall a good result, where if with one measure we see a highly homozygous animal, the correlation implies that it will also be homozygous, highly inbred for all the other measures. So that is good. Here, to answer my third objective of the study, which was to look at historical and recent trends in genetic diversity. First, I estimated the past effective population size in these populations. I used this GON software, and I calculated the effective population size all the way from 5 generations ago. So I looked at animals born in 2020 and 5 generations ago, using where 1 generation equals to 5 years, right? We see that it is from 1995. So from 1995 all the way to 100 generations ago, which is around the 16th century. So I got estimates for all the 5 breeds in all of that time. So let me dive in a little deeper. Here, I show the estimates from 21 generations ago all the way to 100 generations ago. So I want you to notice several things. The breeds, the trends in the effective population size have been different in all the breeds, and there is several reasons why. We can see how the Guernsey and the Jersey, which I remind you were created in the Channel Islands, and some historical records have them, the origins of these breeds all the way to the late Middle Ages. So I posit that this effective population size in these 2 breeds, in Guernsey and Jersey, has been small to begin with because the breed has been isolated in this island for the breed creation. We see how the Holstein had a pretty large effective population size all the way up to the mid-1800s, and this coincides almost perfectly with the importation of the first animals to the US from Holland and from Holland to the US, and the establishment of the breed association in the US. The brown Swiss has a similar history. We see since it's brown Swiss has a very high effective population size, but it also drops off right at the same time because these 2 breeds originated in they were imported in the US around the same time, where dairy cattle farming became very prominent and the importation of these European bulls became very fashionable. And Ayrshire has an interesting trend. It starts dropping off around the 1600s and continues to drop off. There are several factors that could be at play here. The creation of the breed in Scotland, the herd bull creation, all importation into the US, etc. So it is less clear, this, because it's very gradual decline as opposed to these bottlenecking events that we see in the brown Swiss and the Holstein population. But we're in the 21st century, so we're interested in what's happening now. I'll get to that in a second. Here are the trends from 5 generations ago all the way to 20 generations ago. We can see all the way to the 1920s, none of the breeds had an effective population size over 200 animals, and it has steadily decreased, right, with these advancements in the selection methods and in the reproductive technologies. We have driven these breeds to low effective population sizes all the way to 5 generations ago. This is a very interesting result. But what is the role that genomic selection is playing for the current levels of genetic diversity? I think this is the most poignant result that I can show you today, so we can get into brainstorming so we can solve this. So What I did, I calculated yearly rates of inbreeding. We calculated yearly rates of inbreeding using the three inbreeding coefficients, and I did it in sires and dams, since these are the animals that are selected and are the animals that will represent less bias of being genotype before and after genomic selection. So I calculated this yearly rate of inbreeding in 3 different time periods. Before the advent of genomic selection, right? From the year 2000 to 2009, I labeled this as the P1 period. During the initial implementation of genomic selection from 2010 to 2014, I separate this from the widespread adoption because not all the breeds adopted genomic selection at the same time. Most notably, Guernsey and Ayrshire adopted a bit later. And then that second period is labeled as P2. It's from 2010 to 2014. And then this last period is after the widespread adoption of genomic selection from 2015 all the way to 2018. Even though I had data for all the animals up to 2020, I only had data for the sires and dams up to 2018. With this effective population size, so with the yearly rate of inbreeding, I calculated the effective population size in these 3 time periods using this known formula. So here are the results for the effective population size in the sires. For the Ayrshires and the Guernseys, although we see some increases from the P1 period, from before genomic selection, all the way to the P3 period after genomic selection, this is most notably for the genomic measures for GRM and RH. You can see, for example, here it increases in the Ayrshires from 0.14 all the way for 0.52% per year, and in the RH from 0.15 to 0.52 as well. And in Guernsey from 0.21 to 0.76 and from 0.24 to 0.60. The unfortunate result was that we didn't have enough animals, especially enough sires and dams for these breeds, which are had a small population size to begin with. So most of our measures were non-significant. So we cannot really conclude anything about these 2 breeds. Here I present the results for the 3 largest breeds for the rate of inbreeding per year. In the brown Swiss, we see some significant increase for all 3 measures from before genomic selection to after the widespread adoption of genomic selection. Yes, we see these increases from P1 to P3. And you can see that the levels are really high, 1% inbreeding per year. But if we turn to the Holsteins, this is more concerning, right? In the P1, we had very low accumulation of inbreeding, 0.03% per year. But in every measure, it has increased from one period to another. For example, here, let's look at RH. From 0.08% per year all the way to 0.97% per year in P2, I got lost. Here, all the way to 1.41% per year. I remind you, these are the elite animals, these are the sires, so they're very heavily selected on. So you will see these really drastic numbers. And in the Jersey, we found some interesting results where in this P1 period, animals were actually decreasing the rate of inbreeding significantly. The Jersey population was becoming more diverse in these first 10 years of these millennia. Why is that? Because in this period, there was very famous, it is noted that a lot of Danish Jersey bulls were imported, these germplasm from Danish Jersey bulls. So, and other populations. So the Jersey population was actually becoming more diverse. But we turn into after the widespread adoption of genomic selection, and the Jersey population has increased from negative rates to positive rates, although it is not comparable to the Holstein and the brown Swiss. Here are the results for the dams. Mostly found similar results than in the sires, albeit to less extremes. In the Ayrshire and the Guernsey, we didn't find significant results, similar as the sires. But in the brown Swiss dams, different from the brown Swiss sires, these differences from the P1 to P3 were not significant. For the Holstein and the Jersey, similar trends happen as in the bulls. We see that the Holstein has increased the rate of inbreeding in the dams from every period, right? And now it ranges from 0.55 using pedigree measures all the way to 0.70 using GRM. And in the Jersey, similar. In this P1 period, we have negative rates, right? Some of them are significant. This one and this one are this one is significant. Negative rates of inbreeding, and then with the adoption of genomic selection, we have gone with positive rates of inbreeding, although not as alarming as in the bulls. With this yearly rate of inbreeding, I calculate effective population size, and here are the sizes for the effective population size in the sires. We can see very, very, very drastic a low effective population size. I remind you, this is the sires, heavily selected population, but still it is below these 50 to 100 animals that we would desire for maintaining variability and decreasing the effects of inbreeding, this inbreeding depression. So it's very, very low for Holsteins, low for Jersey, and very low for brown Swiss. And here are the results for the Holstein, Jersey, and brown Swiss dams. We see that the Holstein dams have similar effective population size than the sires for all three measures. The Jersey dams had very high, this is the population with the highest effective population size in this last period, which is an interesting result to find. And in the brown Swiss, a little bit higher than in the sires, but still pretty low, especially for these genomic measures. So what is the take-home message of my study? First, we found differences across breeds in terms of pedigree and genomic inbreeding levels, this rate of inbreeding accumulation, and the past and present effective population sizes. Overall, the adoption of genomic selection has brought increased rates of inbreeding, as has been seen in other populations, and has decreased the genetic diversity in this breed, as evidenced with the effective population size. This was most notable for the Holstein population. Implication, we still need to dig deeper, we still need more detailed analysis, fine-tune how we measure genetic diversity so that we can properly census the genetic diversity in these breeds. And I put US, but worldwide populations. And we need to come up with more intelligent strategies to curb this accumulation of inbreeding in these populations. We need to work with farmers, we need to work with breed associations, and we need to put our heads together and come up with more detailed measures that can maintain the genetic diversity that they actually have and decrease the negative effects of the inbreeding. Thank you for your attention, and I welcome any questions. Thank you.
I will use your microphone.
Huh?
I will use your microphone.
Sí, sí.
We share.
Sí.
No COVID. There is a question from the audience. Gustavo asked you if, according to you, the FGRM is the best way to estimate inbreeding.
The FGRM has a very high correlation with RH. I think, as like Christian mentioned, as we get more, we fine-tune these measures, the RH inbreeding will come out on top. Just because we're measuring these, we're able to differentiate between recent and ancient inbreeding. We're able to localize inbreeding depression into certain genomic regions. And overall, it tracks autozygosity much better than GRM, which is a measure that combines IBD and IBS, homozygosity. So overall, right now, if you are able to implement measures that curb inbreeding accumulation based on GRM, that will do a good job, better than the pedigree inbreeding. But overall, we should strive towards using RH and measures such as Christian had shown. There's model-based measures that all are being developed and fine-tuned that can be used. So I think this is the future. better methods, better detection of inbreeding, and better detection of inbreeding depression.
Is there any other question? How do you use that?
If I understood well, I'm not sure. You show that the Jersey population at the beginning had a lower inbreeding rates because of using Danish bulls. Does this mean, according to you, that the Danish Jersey and the US Jersey are low inbred, low related populations, or used to be maybe in the past?
I think Heather Hewson from Cornell has a study on that, about the kinship between the different populations of jerseys. I wouldn't say, but according to the results that I found, yes, they were differentiated enough that when you brought this population from this germplasm from Denmark or from other countries, just due to the selection differences in the indexes, you will increase diversity and decrease inbreeding. Thank you. Any other questions? Thank you very much.